all well? We started the fourth chapter, molecular basis of inheritance. Today we discuss about protein synthesis. Protein is most important component in our body. Protein is a biomolecule, and that protein synthesis takes place. The formation of protein. प्रोटीन तयार होण्याची क्रिया ही कुठे घडून येते? प्रोटीन सिंथेसिस टेक्स प्लेस in the cell of our body. Protein synthesis takes place each and every cell of our body. And the protein synthesis process, there are two steps includes there, that is transcription and translation. After that we discuss independently or separately, that is transcription and translation process. But proteins is work as a enzyme. Protein हे enzyme म्हणून पण काम करतात. Protein work as a hormones also. And protein is a structural component of the cell. That proteins, which place that protein synthesis takes place? In cell. This is the cell, and in cell there are nucleus, and in that nucleus chromosomes are present. These chromosomes, structure of chromosomes here, and in that structure of chromosomes there are DNA, coiling DNA are present, and that DNA present the nitrogen based sequences or present nucleotide sequences. Nucleotide sequences. Nucleotide sequences or nitrogen based sequences. The nitrogen based sequences provide the platform of protein synthesis. That nucleotides or nitrogen based sequences provide the platform or provide the place of protein synthesis. In protein synthesis there are central dogma is the enzyme that help in the transcription process. DNA पासून mRNA तयार होतो, त्या प्रोसेसला ट्रान्सक्रिप्शन म्हणतात. And in prokaryote DNA कुठे प्रेझेंट असतात किंवा eukaryote, we know that eukaryotic cell, and in that eukaryotic cell nucleus, in nucleus DNA is present here, but prokaryote DNA is present on the nucleoids. In prokaryote DNA present in nucleoids, in eukaryote DNA present in the nucleus here. DNA synthesis, protein synthesis takes place in the cell cycle, that is the G1 and G2 phases here. G1 and G2 phases are the growth phases of the cell cycle. In that phases the protein synthesis takes place. Then the next which is the transcription process, there are promoter site, terminator site. That promoter site which start the transcription process, termination site that stop the transcription process here. Then the transcription unit. What is transcription unit? Each transcribed segment of DNA, that is transcription unit, which is the segment of DNA. Suppose it is a DNA and the transcription unit, this is suppose the segment of DNA, this is the transcription unit. Transcription unit of DNA, that is called a transcription unit. And transcription unit has important process, that is the promoter, structural gene and the terminators. Then here the transcription unit, the unit consists here that three parts, that is the promoter, structural gene and the terminators here. Here DNA has two strands, that is strand has the opposite polarity, that is one strand which is fifth to third direction and another strand which is third to fifth direction. In that the promoters bind to the fifth direction of the coding strand. Here these two DNA strand, that one DNA strand which is the coding strand, coding strand, coding strand, that DNA sequences or the nucleotides that are code to code for protein synthesis. प्रोटीन सिंथेसिस तयार करण्यासाठी ज्या स्ट्रँडचे न्यूक्लियोटाइड सिक्वेन्स किंवा न्यूक्लियोटाइड्स हे कोड केले जातात त्याला कोडिंग स्ट्रँड असं म्हणतात. किंवा ह्याच्या सारखंच दुसरं mRNA स्ट्रँड तयार करायचं त्याला कोडिंग स्ट्रँड असं म्हणतात. and one another strand or remaining strand which is the template strand, template strand, that promoter side bind to the fifth direction or fifth side of the coding strand. It is a promoter, promoter, and the terminator bind to the third end of the coding strand. Terminator bind to the third end of the coding strand, terminator. Here promoter bind to the fifth end of the coding strand, terminator bind to the third end of the coding strand. One strand which is the template strand and another strand which is the coding strand, and then the remaining part which is the structural gene, structural gene. Structural gene. Here what is promoter? The promoter which is the small sequence of nucleotide or small sequence of DNA that provide the binding site for the RNA polymerase, that provide the binding site. RNA polymerase bind करण्यासाठी ची जागा जो देतो तो कोण आहे? promoters आहे. and which is the small sequence of DNA. promoter हा काय आहे? छोटा sequence डायरेक्शन असते. म्हणून हा जर कोडिंग स्ट्रँड असेल तर नवीन mRNA स्ट्रँड हा कोण तयार करणार आहे? दिस स्ट्रँड, दॅट इज द टेम्पलेट स्ट्रँड which transcribe the new mRNA and that new mRNA here and that direction which is fifth to third. Because of the template strand direction which is the third to fifth, we here discussed that is the transcription unit, and in that transcription unit, that is template strand, coding strand, promoter, structural gene and the terminator and the promoter factor and the terminator factor. Then we discuss here the two strand of DNA. One is the fifth to third direction and the second is the third to fifth direction. We know we discuss about the fifth to third direction which is the coding strand. If it is coding strand and it is template strand, it is template strand. And the sequences of the nitrogenous base or the pairing of nitrogenous base here or pairing of nucleotides here, A to T here. Another strand of DNA, it is one strand, it is another strand. G, again pairing C, A, T, A, C, G, A, T, then G, C, T, A, T, C, G, A, T, T, A and G which is the C. Here the DNA nucleotide sequences and which is present at the nucleotide sequences on the template strand and the coding strand here. Here the pairing of nitrogenous base here. A always pair with T, G always pair with C or C always pair with G. Then T always pair with A, C always pair with G, A always pair with T here. It is a nitrogenous base pairing here. And in this here, when we, it is a coding strand. परंतु ज्यावेळेस पण आपण ह्या कोडिंग स्ट्रँडचा जर ट्रान्सक्रिप्शन साठी वापर केला तर काय होते बघूयात आपण. कोडिंग स्ट्रँड when we transcribed in mRNA here or RNA, that thymine is replaced by uracil, that T is replaced by U. Then here A which is adenine which is not replaced or adenine is as same in DNA in RNA. Then guanine, cytosine which as same in DNA and RNA, but thymine is replaced by uracil here. When we here जर आपण ह्या कोडिंग स्ट्रँडचा वापर केला ट्रान्सक्रिप्शन साठी, you here keep in mind and keep in remember here, जर आपण कोडिंग स्ट्रँडचा वापर केला mRNA तयार करण्यासाठी तर mRNA कशा प्रकारचा तयार होते बघा. समजा हा mRNA इकडे जरी लिहिला किंवा इकडे जरी लिहिला, आपण इकडे लिहूयात. समजा mRNA इकडे लिहूयात आपण, दॅट इज इथं आपल्याला माहित आहे नायट्रोजनस बेस सिक्वेन्स असताना A हा कुणा बरोबर पेअरिंग करतो? T बरोबर पेअरिंग करतो. परंतु आपल्याला mRNA तयार करायचा आहे. mRNA तयार करत असताना A बरोबर T पेअरिंग होत नाही, तर T च्या जागी काय रिप्लेस आहे RNA मध्ये? T च्या जागी uracil U, दॅट इज A च्या जागी U, G च्या जागी हियर C small nuclear RNA, snRNA. Our transcription process which helps the RNA polymerase